would like to start with an apology for being a little late to join this session. Murphy's Law caught up with me. My computer was throwing tantrums, and fortunately I was able to put together an alternate system, and I hope that it lasts for the next 1 hour or so until we finish the session. So let us get started right away. I would just like to point out that this is the first session of a series of webinars that has been planned as part of this neurophysiology series. So we will basically be looking at introducing basic neurobiology for psychiatry postgraduate students. I understand that the audience for this are psychiatry postgraduate students. The perspective broadly would be a cognitive neuroscience perspective, because that's where a psychiatrist's interests would lie. There are a couple of kind of caveats or disclaimers that I would like to share with you in the beginning of the series itself. This is going to be, these presentations, these discussions are going to be quite selective. There is no intention or attempt to make it exhaustive. In a 6-hour series, neuroscience can only be skimmed on the surface. Secondly, the focus is on broad principles. It is not on technical details. Having said that, at the risk of sounding a little, at the risk of making this a little possibly irrelevant, I would like to keep the focus on the neuroscience, the basic neuroscience, because from what I've seen, there is more than sufficient opportunity for applying principles to clinical understandings that psychiatry postgraduates have. But because of paucity of time, often we do not have enough time to develop on the basics of neuroscience. So I'm going to restrict myself to the basics of neuroscience. So these are the kind of caveats or disclaimers that I would like to point out at the beginning itself. So moving on, to set up this series, I just want to remind you all that we can understand brain function at multiple levels. Of course, as psychiatrists, you would be most interested in behavior and, of course, the mind. I think we are beyond debating now that amongst neuroscientists that mind and behavior are the outcomes of what the brain does, and therefore the highest level at which one can study, of course, is the brain and insights into mind, and then, of course, the brain as a whole, functioning of the brain. So there are various approaches to look at brain functioning as a whole. The most popular ones currently are, functional imaging, electrophysiology, magnetoencephalography, etc., as far as brain functioning is concerned. And of course, looking at the structure of looking at the microscopic structure of the brain is possible, at least in postmortem studies. At the next level, we have the systems level. For example, you would have the visual system level, the auditory system, the learning and memory systems, the speech comprehension and speech production system, the various systems involved in emotional regulation, mood, etc. So these would be multiple areas of brains networked together, working together to bring about a specific function. One step below that would be the microcircuitry level or the local circuitry level. So, for example, within the visual cortex, you have neurons forming complex circuits to bring about visual function. Within the hippocampus, you have very well-studied, increasingly well-studied circuits which support learning and retrieval of memories, and so on and so forth. So this is the circuit level. If one goes below that, you would have the neuronal level. And of course, below that, you would have the synaptic circuits and the synapses. So the neurons and synapses are, of course, a very important element of one can consider the neurons and synapses as the functional units of the brain and therefore of neuroscience. And one would need to relate the functioning of neurons and synapses to circuits and then to systems and then to brain, and of course to the brain and therefore the behavior. So this is how one kind of drills down different levels of study, different levels of understanding. You could go down to molecules like ion channels or receptors, and even down to genes. And there is even a fledgling field of quantum neuroscience, which would even go down to subatomic levels. So one can study neuroscience at multiple levels, and we would in this series would try to make these connections, especially starting from synaptic and neuronal level right up to the brain level, and touch upon the behavioral and psychological correlates of this. So this would be the approach by and large in this series. In the coming weeks, we would explore 4 major areas of brain functioning at these multiple levels, from synaptic neuronal level right up to brain level. These would be the learning and memory function, the fear and anxiety related to emotional regulation, sleep, and decision making. So these are the 4 major domains of brain functioning that we would be looking at in the coming weeks. What is important for all of these, which would bind all of these together, is neuronal signaling. And therefore we start with neuronal signaling today. What are the fundamentals of the biological basis of brain functioning? The obvious answer would be the communication between neurons, and that's what we would focus on. We would, in today's session, look at neurons, the role of neurons in signaling and in integrating information. We would start off with that. We would look at membrane potential changes as the basis of neuronal signaling. So we look at things like excitability, local potentials, and action potentials. This is something that we learn right in first MBBS, and then we kind of put it in the back burner and forget about it. So I would spend a little time on this because it is an excellent example for the multidisciplinary nature of neuroscience, which has brought together scientists from varied fields of science and even humanities, and the kind of progress that has been made in the last century is simply amazing. And then we would move on to signaling between neurons, focusing on synaptic transmission, look at how EPSPs, excitatory and inhibitory postsynaptic potentials are generated, touch upon neurotransmitters and postsynaptic receptors, and then end up with how intracellular signaling pathways play a role in neuronal functioning and survival. So the intraneuronal signaling. So this is the outline of today's session. I'd like to begin with a very well understood, a very familiar phenomenon, the stretch reflex, which actually kind of provides the most basic unit of understanding neuroscience. So if you stretch the tendon of a muscle, like for example the quadriceps muscle, with a quick tap on it using a knee hammer, it would lead to activation of stretch receptors within the muscle. This information about the stretch of the muscle is conveyed into the spinal cord through a single neuron, the spinal afferent 1A spinal afferent neuron, which forms a direct connection with alpha motor neuron, which in turn sends out an axon through the anterior root and through a nerve, and activates the muscle fibers of the quadriceps muscle to bring about contraction. So look at this extremely simple neural network. It is basically a 2-neuron network, one afferent neuron and one efferent neuron, and they communicate within the anterior horn of the spinal cord. And we have all aspects of the fundamentals of neurobiology happening here. Sensation, converting that sensation into a signal, transmitting that signal through a neuronal axon, conveying that information into a second neuron. The second neuron integrates that information and then sends out an action signal, again in the form of an action potential, to the effector organs, in this case the muscle fibers. So everything else that the brain does is simply elaboration of this, adding layers of complexity to this. At the end of the day, like the famous physiologist Charles Sherrington almost 100 years ago said, all that we humans can do is move muscles or secrete through glands. and that's what the brain does. And so therefore, it is interesting to begin with this perspective. If you reduce this 2-neuron circuit, we can look at the steps of the basics of the neurobiology of the brain. So this is an afferent neuron on the left side communicating with the cell body of the alpha motor neuron on the right side. So a stimulus, which could be a stretch, in the case of stretch reflex, it could be a change in temperature, it could be a ligand binding there, a specific ligand binding to receptors on the receptive end of the neuron, or it could be any other form of stimulus, which would lead to formation of a small change in the potential in the local area of the stimulated part of the neuronal membrane. So this local potential, if it is large enough, will lead to generation of a massive change in membrane potential called as the action potential. And this action potential would travel down the neuron to the other end of the neuron, where in this case it would reach a synaptic terminal on the alpha motor neuron. And once this action potential reaches the synaptic terminal, it leads to release of neurotransmitters from this terminal into the gap between the synaptic terminal and the alpha motor neuron membrane. And these neurotransmitters would act on the postsynaptic membrane on the alpha motor neuron, and they would lead to activation of the alpha motor neuron. Now, the interesting thing is, if the receptor potential, if the stimulus results in a larger receptor potential, you instead of a few action potentials, you have a burst of action potentials. In other words, higher frequency of action potential generation, and these action potentials would travel down and they would lead to release of more neurotransmitters. So we have the basis of encoding the strength of stimulus here. Larger stimuli would lead to larger bursts or more frequent bursts of action potentials, which would lead to more and more release of neurotransmitters. And ultimately, the amount of neurotransmitters acting on the potential in the postsynaptic side, and therefore the communication chain continues. The signal has been successfully transmitted. So this is the basis of any brain function. This is how neurons function in chains or networks. Everything else is basically an elaboration of this. So what do neurons do? At the end of the day, what do neurons do? Neurons sense, in the sense that, pun intended, in the sense that they sense changes and cause transduction. That is, they convert a touch or a light signal or a chemical signal into a neuronal signal. These changes in membrane potentials in the receptors are translated into action potential. This is signal encoding, and this is how information is generated. Then you would have the next step where neurons play a role is sharing this information. So they share it with other neurons. So synaptic transmission plays an important role in this transmission of information from neuron to neuron. And this is a hugely complex area of neuroscience. And then you have the processing of information. Neurons receive inputs from multiple neurons, and they help in integrating these inputs, in associating in these inputs, and ultimately this is how deductions are made about the inputs and transmitted on to the next in-chain neurons. Information can be stored by networks of neurons, not individual neurons, but networks of neurons, and this information can be retrieved, and this forms the basis of learning and memory. Neurons can generate action signals, they can formulate action plans, and they can implement action plans through motor neurons, and this forms the basis of action of the brain. And neurons, of course, activate muscles or glands and bring about behavior. So everything that the brain does it is able to do because the neurons support it in sensing, encoding, sharing, processing, storage and retrieval, generation, and activation. There are different ways of looking at what the brain does. One attractive way of looking what the brain does is basically the brain has evolved to act. And therefore, everything that the brain does, collecting information, integrating information, and sending out action signals, is everything related to the brain's role in acting. So, in that sense, action can one perspective is that action is the basis of everything, or is at the center of everything. It is important to realize that single neuron by itself is insignificant, and therefore I've underlined the S in neurons. It is neurons which work together as networks which are able to do everything that the brain does. So neurons have charged membranes. We'll look at this in a bit of detail later on. And changes in this charge distribution or changes in the potential is used as a signal, and membrane potential changes forms the basis of neuronal communication. Neurons do not work in isolation, like I said. The simplest of brain functions require networks of neurons, and these networks are formed through connections called as synapses. A very interesting and very important aspect of neuronal function is that neurons are changed by their connections with other neurons, and vice versa. Neurons change the neurons to which they are connected. This is an extremely important aspect of brain functioning, that neurons work in networks, neurons affect other neurons, the way neurons are connected to other neurons changes over time, and neurons are affected or changed by other neurons. And therefore, we are going to look at three aspects today. I'm really going to fly through some of these things because some of these things are quite basic, but still I would like to fly through these. Maybe about 15 minutes for part one, where we look at membrane potential changes as the basis of neuronal signaling. Then we move on to part two, which is synaptic transmission, the communication between neurons. We'll focus on chemical neurotransmission. And the last part is the intraneuronal signaling. So I'm warning you, this is going to be almost a supersonic flight. Part one, membrane potential changes as the basis of neuronal signaling. If you insert a recording electrode into a neuron, before that electrode pierces the neuron, you have no potential recorded across the membrane. That means there is zero potential across the membrane. Once a recording electrode pierces the neuron, the electrode records a negative potential. This negative potential remains stable as long as the neuron is unperturbed. And therefore, we call this as a resting membrane potential. If that electrode is withdrawn, that means there is no potential difference between the two electrodes, recording and reference, and therefore there is a zero potential recorded. So the bottom line is that the interior of the membrane is a negative with respect to the exterior of the membrane in most cells, in almost all mammalian cells, and this we call as resting membrane potential. The difference between interior minus exterior. Different cells have different resting membrane potentials, but they're all negative. Epithelial cells, for example, have a minus 40 millivolt difference. Neuronal cells vary between minus 70 millivolts to minus 90 millivolts. Minus means interior is negative with respect to outside. Cardiac cells and muscle cells also have about minus 90 millivolts of difference. So the membrane is polarized, and the polarized membrane how does the membrane become polarized? What do we mean by polarized? Basically, what we mean by polarized membrane is that the charge separation occurs. There are more negative charges on the inner side of the membrane compared to the outer side. How does this happen? A simple thought experiment helps us to understand this. If you have a semi-permeable membrane that is permeable only to potassium and not to chloride in this case, separates two compartments with a higher concentration of potassium chloride on the left side and a lower concentration on the right side. And if you are recording potential difference between the left side and the right side, you would not expect to see any potential difference because the positive charges and the negative charges are balanced on either side of the semi-permeable membrane. However, if the membrane is made permeable to potassium, then what happens is, because there are more potassium ions on the left side compared to the right side, potassium ions would diffuse down this chemical gradient, and some potassium ions would move to the right side, adding positive charges to the right side and leaving behind unaccompanied chloride channels, and therefore a buildup of negative ions on the left side. And therefore, what we see is that the chemical gradient drives ions down this membrane, and because of that, you have more positive charges on the right side, less or more negative charges on the left side, and therefore charge separation occurs. And therefore, what you see is a recording of a potential difference. So the bottom line here is that a chemical gradient difference in ion concentration has resulted in an electrical gradient. And this movement of ions, of potassium ions in this case, from left to right, will continue until the electric gradient becomes strong enough to oppose the chemical gradient. And that the potential level at which this happens is called as the equilibrium potential for potassium. So the electrical gradients result from movement of ions driven by chemical gradients, and that potential at which the chemical gradient is effectively opposed by the electrical gradient is called as equilibrium potential. So what determines the magnitude of the equilibrium potential for various ions? Simple thing, if the concentration difference is greater, you end up with greater charge separation and therefore greater equilibrium potential. The second thing is, of course, the nature of the charge. The direction of the ionic gradient, electrical gradient, is determined by whether the ion is positive or negative. So Nernst, way back in 1908, put all of this together and gave a very straightforward formula, which says that the electrical potential established across a membrane because of differences in ion concentration is determined by the concentration difference and the nature of the charge, positive or negative, and you can have formulas to determine this. Now, the thing is, does this help us in understanding resting membrane potential? Now, if electrical gradients results from movements of ions driven by chemical gradients, we can relate this to the cell membrane. Neuronal membranes have differences in the concentration of ions in the intracellular fluid on the intracellular side versus the extracellular side. For example, there's a lot more sodium on the outside compared to inside. There is much less potassium on the outside compared to inside. Calcium is 5 to 1 times or 10 to 1 times more on the outside compared to inside, and so on and so forth. So, and the cell membranes, the neural membranes, are not equally permeable to all ions. So we have a situation where there is a concentration difference of ions across the cell membrane, and the cell membrane is semi-permeable. It is selectively permeable, and therefore we have the basic requirements for setting up of electrical gradients driven by chemical gradients. So cell membranes are essentially non-permeable. The lipid bilayer is non-permeable, but protein channels and protein transporters which span the cell membrane allow ions to cross the cell membrane and therefore provide selective permeability to cell membranes. The ion channels are basically pores, protein-lined pores, which allow the free movement of ions across them, through them. And there are essentially two types of ion channels: the non-gated channels, which are always open, and the gated channels, which are sometimes open and which are sometimes closed to ion movement. So the closed channels, the gated channels may be in an open state or a closed state. And the closed channel can become open either when the membrane potential changes. These are called as voltage gated channels, or when a ligand binds to it, a chemical substance binds to it and kind of opens a lock. These are called as ligand-gated channels. And you also have mechanically gated channels. A stretch can lead to opening of channels, or even temperature-sensitive channels. So coming back to our cell membrane, because of the concentration difference of potassium, sodium, chloride, and other ions, we can think of equilibrium potentials for each of these ions. For example, if you use Nernst equation, you can determine that for a spinal motor neuron, the equilibrium potential for potassium across the neuronal membrane is about minus 90 millivolts. For sodium, it's about plus 60 millivolts. And for chloride, it's about minus 70 to minus 90 millivolts. So if you simplify the cell membrane to a simple diagram, and if you imagine that the cell membrane is permeable only to potassium, then based on our previous logic, potassium should diffuse from inside to outside until charge separation occurs and the electrical gradient develops, and until this electrical gradient becomes large enough to oppose the movement of potassium ions. Now, the electrical gradient builds up to about minus 90 millivolts. That is, the inside becomes negative compared to outside by minus 90 millivolts. Only then it equilibrates, it opposes the movement of ions, potassium ions. And therefore, an important statement I'm making here, if the cell membrane was permeable only to potassium, we would be able to explain the resting membrane potential as the equilibrium potential of potassium, and we would expect it to be minus 90 millivolts. On the other hand, if the cell membrane is permeable only to sodium, then sodium would move from right side to left side, that is from outside to inside, until charge separation is strong enough to result in a opposing electrical gradient to stop sodium movement. Now this happens when the electrical gradient approaches plus 50 millivolts for sodium. That is, outside is positive compared to inside. So if the cell membrane is permeable only to sodium, then the resting membrane potential should have been equal to the equilibrium potential of sodium, which is plus 50 millivolts. But our experiments show that the actual resting membrane potential is about minus 70 millivolts. Therefore, it is neither equal to that of potassium, equilibrium potential of potassium, nor is it equal to the equilibrium potential of sodium, but it is somewhere in between, but it is closer to the equilibrium potential of potassium. Therefore, putting all this together, the conclusion is that the cell membrane is more permeable to potassium than to sodium, maybe at a ratio of 40:1 in the resting state, and therefore, potassium diffuses from inside to outside to a greater extent, sodium diffuses from outside to inside to a lesser extent, and ultimately the resting membrane potential is pulled towards the equilibrium potential of potassium, but it does not reach the equilibrium potential of potassium. It stops at around minus 70 millivolts. It doesn't reach all the way to minus 90. So sodium does contribute in the sense that it pulls the resting membrane potential weakly towards it, but it is overwhelmed by the strong pull of potassium equilibrium potential, and therefore the resting membrane potential is a weighted average of the equilibrium potential of sodium and of potassium. So one can take this forward and include other ions, and you can have a formula for determining this weighted resting membrane potential, where the weightage is based on the permeability of the membrane to various ions. To summarize, the resting membrane potential originates due to two simple facts. One, that the ions are distributed differentially. The ion concentration is different inside versus outside neurons. Second thing is that the cell membrane is differentially permeable. It is not equally permeable to all ions. In the resting state, it is much more permeable to potassium than it is permeable to sodium. Now, continuing this experiment a little forward, if you introduce a third electrode and inject current, positive current, or withdraw positive current, it's interesting to see that the membrane potential shifts upwards when you inject positive current. This is called as depolarization. If you inject negative current or withdraw positive current, then the membrane potential drops further towards negativity. This is called as hyperpolarization, and such a stimulus is called as hyperpolarizing stimuli. If you give stronger and stronger hyperpolarizing stimuli, you get bigger and bigger hyperpolarization responses. So it's a linear response. On the other hand, also if you give a stronger and stronger positive stimuli, you get stronger and stronger depolarizing responses. Most cells behave this way. These are non-excitable cells where the response, membrane response, is proportional to the stimulus strength. And such responses are called as graded potentials or local potentials. You see this in liver cells, epithelial cells, etc., which are essentially non-excitable cells. On the other hand, if you look at excitable cells like neurons or muscle cells, stronger and stronger negative stimuli, negative current stimuli will lead to proportionally bigger hyperpolarizing responses, that is more negative responses. However, if you increase the strength of depolarizing stimuli, you do get bigger and bigger depolarizing responses. But something interesting happens. The responses become disproportionately larger, not proportionately larger, but disproportionately larger. And in fact, if the membrane potential crosses a particular threshold, you get a very different type of response, a runaway response. The membrane potential becomes very much positive very rapidly. In fact, it crosses zero and becomes positive, and then declines relatively rapidly back to its resting state. And such a response is called as an action potential. And this is characteristic of excitable cells. Neurons, muscle cells, and some other cells can generate action potentials because they are excitable. That means depolarizing stimuli, if they exceed a particular threshold, results in a very different type of a potential, explosive response called as action potential. Action potential has got an initial small, slow response called as local response, and then it's got a rapid depolarization phase, and then it has got an overshoot. Part of it is an overshoot which goes above zero, and then it's got a rapid repolarization phase where it comes back towards the baseline, and then it's got a slow repolarization phase where slowly it returns towards baseline, the resting membrane potential. In fact, it dips a little sometimes after hyperpolarization and then stabilizes at the resting membrane potential. So these are the phases of a typical action potential. If during rest we understand that the membrane is primarily permeable to potassium, and therefore the resting membrane potential is influenced primarily by the equilibrium potential of potassium, what happens during an action potential is that the membrane becomes hugely permeable, massively permeable to sodium for a short time, and therefore the membrane potential is pulled towards the equilibrium potential of sodium. That means towards positivity, towards plus 50 millivolts, and therefore you get an overshoot. But then very rapidly the membrane permeability reverts back to potassium permeability and less permeability to sodium, and therefore the membrane potential returns towards the resting state. So to summarize, we can actually describe the action potential just like we describe the resting potential in terms of changing permeability of the membrane. So increase in sodium conductance followed by a slower increase in potassium conductance leads to the typical changes that one sees in action potential. And we see that when the membrane potential becomes slightly depolarized, it is more likely to become more depolarized, and therefore it sets up a regenerative process, a self-generative process leading to an explosive action potential. So the question that we are left with is, how does membrane conductance change? Especially how does it change during an action potential? The answer to that lies in voltage-gated sodium and potassium channels. The voltage-gated sodium channels are voltage-sensitive channels. They have two gates, one on the inside called as the inactivation gate and one on the outside called as the activation gate. Normally, the activation gate is closed, and therefore they do not allow sodium ions to cross. However, If the surrounding membrane becomes depolarized, then the activation gate opens, and this allows the movement of sodium ions from outside to inside. This is a very rapid action, happens in milliseconds, a fraction of a millisecond, and then you have influx of sodium, which leads to change in membrane potential, which leads to depolarization. Very rapidly, the inactivation gate closes, and therefore this is a very transient opening, and a little later, slowly the inactivation gate opens up, leading to inactivation. The activation gate also closes and returns it to the resting basal state. So this cycle, which happens extremely rapidly, is responsible for the rapid depolarization and overshoot phase of the action potential. You have potassium-gated channels which have a single gate on the outside. Again, membrane depolarization leads to opening of this channel, but it's a much slower response. These channels begin to open when the sodium channels are actually beginning to close, and this allows efflux of potassium, outer movement of potassium, which leads to repolarization of neuronal membrane. And then later on, it returns to basal state. Therefore, one can look at action potential generation as a regenerative process where initially there is increased sodium conductance and then there is increase in potassium conductance, leading to the typical action potential. During the rapid depolarization phase, you have sodium influx through opening of voltage-gated sodium channels, and then during the repolarization phase, there is efflux of potassium through voltage-gated potassium channels. Now, when the stimulus is subthreshold, it leads to generation of a small potential change, which actually rapidly dies down, and also it is transmitted only to short distances because it dies down as it's transmitted. So these are called as local potentials or electrotonic potentials, whereas a stronger stimulus leads to generation of an action potential, which is transmitted rapidly, which builds up rapidly, and which is also transmitted without much change. That means the amplitude remains the same. It is transmitted rapidly to long distances. Therefore, these are the fundamental differences between the passive local potentials and the active action potentials. Passive potentials are local graded. They are proportionate to the input stimulus, whereas action potentials are spiky in nature, and they are all are phenomenon. They are stereotyped potentials. Local potentials are conducted electrotonically. They're decremental. That means they die out quickly. Action potentials do not die out quickly. They're regenerative. They can be transmitted long distances. There are other properties also which we will not discuss today. Now, ion channels are of various types. They allow movement of different ions. They all belong to broadly three superfamilies. The glutamate type of channel family, then you have the gap junction type of family, and you have the voltage-gated sodium channel type of family. So almost all ion channels belong to one of these three categories of channels, which are coded by three superfamilies of genes. Now, it's important to understand that membrane potentials are sculpted by ion channels. Depending upon the type of ion channels and the density of ion channels, the excitability of neurons differ. Also, the size, shape, and frequency of action potentials differ. We have examples of different types of neurons here. You have a regular spiking neuron here, which kind of regularly fires action potentials. In B, you have a fast spiking neuron. Again, it's regular, but the rate of firing is much higher compared to the regular spiking neuron. And you can also see that depending upon the stimulus intensity, the frequency of firing of action potentials changes. In C, you have an intrinsically bursting neuron, which gives bursts of neurons initially and then gives sparse firing of neurons. You have chattering neurons, which you have multiple bursts of neurons. So different neurons respond differently. Their action potential types and frequencies and the clustering of action potentials is different. And all this is because of the differences in types of ion channels that they possess. So to summarize, membrane potentials can be passive, local, or regenerative in action and transmitted. Regenerative action potentials happen through voltage-dependent changes in conductance. They're suited for long-distance signaling. They trigger neurotransmitter release in synapses. Local potentials are suited for neuronal integration, and they do not travel long distances. So with that, we come to the end of part 1. I'm going to spend just about 5 minutes on part 2, signaling between neurons, where we will look at synaptic transmission, which provides robust and modifiable mode of interneuronal communication. So we'll quickly look at CNS synapses. There are broadly 2 types of CNS synapses. Synapses are the functional junctions between neurons. These are regions of contiguity, not of continuity. That means there is a clear separation between where one neuron ends and the other neuron begins. So there is a synaptic cleft which separates the membranes of the presynaptic neuron and the postsynaptic neuron. The type 1 neurons usually form on dendrites. These are characterized by dense postsynaptic specializations, and the synaptic cleft is a little wide. Type 2 neurons, which are axosomatic, that means on the cell bodies, or on the axons, that is axo-axonic. These are they have flattened synaptic vesicles as compared to spherical synaptic vesicles in type 1. Their postsynaptic densities are less prominent, and these are often inhibitory in nature. All of us know the basic steps of synaptic transmission. I don't need to really dwell much upon this. The arrival of a action potential in the presynaptic terminal leads to opening of calcium channels. This leads to entry of calcium through this voltage-gated calcium channels. The calcium triggers a series of events within the presynaptic terminal, which leads to the docking of synaptic vesicles with the synaptic membrane and to the release of neurotransmitters. The neurotransmitters in turn diffuse across the synaptic cleft and act upon postsynaptic receptors on the postsynaptic membrane. And these postsynaptic receptors could be channels which open, therefore allowing movement of ions. When you have movement of ions, it changes membrane potentials. It either depolarizes neurons or hyperpolarizes neurons, and therefore you have excitatory postsynaptic potentials if it is a depolarizing response, or inhibitory postsynaptic potentials if it is a inhibitory response. So these are the basic fundamental steps of synaptic neurotransmission. What happens within the presynaptic terminal at the arrival of a action potential? Like I said, voltage-gated voltage-gated calcium channels in the presynaptic membrane open up, which leads to entry of influx of calcium. Now, this leads to the binding of fusion of synaptic vesicles and exocytotic release of neurotransmitters. Each synaptic vesicle contains thousands of neurotransmitter molecules, and these synaptic vesicles are recycled. That means after some time, there is a pinching in of the synaptic membrane, which was part of the synaptic vesicle, which had fused with the presynaptic membrane. And this is a kind of reversal, a kind of an endocytotic process, which leads to formation of a reformation of a vesicle. So there is a recycled vesicle, and these vesicles are filled with neurotransmitters. So there are specialized proteins which help in pumping neurotransmitters back into these vesicles, and they become available in the pool of synaptic vesicles for release once again. So all these are targets for drugs. And these are all possible sites at which things can go wrong, leading to neuropsychiatric dysfunction. So you could have problems with voltage-gated calcium channels. One can have problems in the docking of synaptic vesicles and the triggering of neurotransmitter release. There could be problems with the pumping of neurotransmitters back into vesicles. Drugs can affect this. For example, there are a class of drugs important in psychiatry which inhibit the reuptake of neurotransmitters into vesicles and increase. There are a lot of experimental drugs which could potentially interfere with these processes. There is an increasing amount of information about how actually docking of synaptic vesicle happens. For example, there are proteins called as syntaxin and SNAP25 proteins on the cell membrane, and there are proteins called as there's a protein called as synaptobrevin in the synaptic vesicle. Apparently, synaptobrevin interacts with syntaxin to kind of anchor synaptic vesicles to the presynaptic membrane, and then the availability of calcium leads to fusion of the synaptic membrane with the presynaptic membrane, vesicular membrane with the presynaptic membrane, leading to exocytosis of transmitters. There is also increasing knowledge about how calcium plays a role here. Calcium binds to a protein called as synaptotagmin, which is part of the docking complex. And once calcium binds there, there's a kind of a unzipping of this vesicular membrane and fusion of the membrane with the presynaptic membrane. As you can see here, it modulates these proteins like syntaxin and synaptobrevin, and therefore synaptotagmin is an important molecule which interacts with calcium and brings about these effects. There are various types of neurotransmitters broadly classified into two types: the small molecule neurotransmitters and the large molecule or the neuropeptides. The small molecule neurotransmitters, one important category is acetylcholine in itself. Then you have the amine, biogenic amines like dopamine, norepinephrine, epinephrine, serotonin, histamine, etc. Then you have the amino acids like aspartate, glutamate, glycine, GABA. All these. Therefore, a large number of small molecule neurotransmitters are ultimately derived from amino acids. Whether they're amines or amino acids, they're derived from amino acids. So, amino acids are important precursors for small molecule neurotransmitters. ATP, adenosine triphosphate, is also recognized as a small molecule neurotransmitter. The other major class of neurotransmitters are the peptide neurotransmitters. You have multiple families here: the opioid family, including opiocortin, enkephalins, anandamide, etc. Hypophyseal neuropeptides like vasopressin, oxytocin, etc. Tachykinins like substance P, substance K, bombacin, etc. Secretins like secretin, glucagon. So many of these are actually gastrointestinal peptides. They have a role in gastrointestinal physiology too. In the brain, they have important neuromodulatory functions. Insulin and insulin-like growth factors are important, recognized to be important transmitters, especially involved in survival and growth of neurons. Then you have somatostatins, including pancreatic polypeptide and gastrins. So a large number of peptides have double roles in the GIT as well as in the brain. In the brain, they act as neuromodulators. Neurotransmission, synaptic transmission can be fast or it can be slow. The fast excitatory synaptic transmission is most commonly mediated by glutamate, by its action on inotropic glutamate receptors. There are two types of inotropic glutamate receptors, the AMPA receptors and the NMDA receptors. Both of them allow sodium and potassium to cross them. So they are cation selective receptors. Both AMPA receptors and NMDA receptors are very important in neuropsychiatry. We will look at the role of these receptors in synaptic plasticity, in learning and memory, and in other aspects of neuropsychiatry in the subsequent sessions. The Nmda receptor in particular is interesting because it is not only modulated by membrane voltage changes, but it also is modulated by availability of other substances like magnesium, glycine, etc. And therefore, it really plays an important role in integrating multiple inputs into neurons. The other class of glutamate receptors are the metabotropic glutamate receptors, which we'll study in greater detail later. They mediate slower transmission, slower neuromodulation. The fast inhibitory synaptic actions are mediated by inotropic GABA receptors and glycine receptors, both of which allow chloride ions to pass through them. We'll look at these again a little later. So synaptic inputs, neurons receive multiple synaptic inputs from multiple neurons, and neurons have an integrative function. That means inputs which occur either within a short time or which occur close together on the neuronal surface can be added together. That means if you have two excitatory inputs given one after the other within a very short interval, then the postsynaptic neuron integrates them, adds them together to form a bigger response, and therefore a higher frequency of action potentials. So this is called as temporal summation. Similarly, two synaptic inputs which are spatially close together or which are occurring on the same neuron can be integrated together through spatial summation, leading to larger local responses and ultimately to higher frequency action potentials. So if you have positive excitatory inputs and inhibitory inputs, it's kind of algebraic summation. It becomes the sum of the excitatory plus the sum of inhibitory. So if the total net excitatory inputs are greater than the net inhibitory inputs, you end up with a net excitatory effect and vice versa. So synapses allow for integration of information from multiple sources. So to summarize this part 2, synaptic transmission, the action of a neurotransmitter depends on the properties of postsynaptic receptors. This is very important. It is not really dependent upon the neurotransmitter, but rather on the postsynaptic receptor. For example, dopamine can act on one set of dopamine receptors to cause excitatory postsynaptic potentials. The same dopamine, exactly same neurotransmitter, can act on a different set of postsynaptic receptors to cause an inhibitory postsynaptic potential. Therefore, what is important is the nature of the postsynaptic receptor, which that is what determines whether inputs are ultimately excitatory or inhibitory in nature. Activation of postsynaptic receptor gates or ion channels directly or indirectly underlies the ability of neurotransmitters to modulate the excitability of neurons. And central neurons, of course, can receive inhibitory or excitatory responses and integrate them. Excitatory responses are most commonly mediated by inotropic glutamate receptors and inhibitory most commonly by inotropic GABA and glycine receptors. So that is a summary of synaptic transmission. We will revisit many of these things in the context of learning, memory, anxiety behavior, etc. So I'm only touched upon the fundamentals today. Now, part 3, which is intraneural communication, that is postsynaptic receptors recruiting multiple intracellular pathways to modulate membrane excitability, neuronal activity, and survival, which is essentially neuromodulation. Now, what I propose is, because we have already run out of time today, and we also lost a little time in the beginning, what I propose is, I will deal with this part. I will introduce you to this part as part of a session on the neural basis of learning and memory, because it is actually becomes relevant there. I need about 10 minutes for this. So instead of dragging on today and making it rather unbearable, because of the nature of the topic, I will introduce this to you in the session in the neural basis of learning and memory. That would be possibly the third session. And therefore, I will not discuss this today. What I'll do is, I will quickly summarize what we discussed today. So what we discussed today is that one, neurons are the fundamental functional units of the brain. Neurons play a role in sensing, encoding, transmission, integration, storage, and retrieval of information. They also play a role in generating intention for action, action plans, and actually recruiting effector organs like muscle fibers and secretory cells. Second thing, we saw that the basis, biological basis of the ability of neurons to do all that they do is their ability to modulate their membrane potential. The membrane potential is nothing but charge separation across the membrane, and this occurs because of three fundamental factors. One, the membrane is differentially permeable to ions. Two, the distribution of various ions across the membrane is different. And the third part, actually a small part, that's why I didn't spend any time on that, is the role of the sodium-potassium pump, which is an electrogenic pump. Once you have a membrane potential set up, this membrane potential can be modulated. Membrane potentials can become more positive or less positive, that is more negative, either by the action of chemical substances which act on ligand-gated ion channels, or through voltage-gated ion channels which sense voltage changes and open or close, or through mechanical action, through stretch of the membrane leading to opening or closing of ion channels, or also through temperature changes. So changes in ion channel opening, closing leads to changes in membrane potential, which leads to local potentials and action potentials. And this is the basis of all neuronal action. Third, we saw synaptic transmission, where we have a series of presynaptic events and postsynaptic events where neurotransmitters are released and act on postsynaptic receptors. And depending on the nature of postsynaptic receptors, you have excitatory postsynaptic potentials or inhibitory postsynaptic potentials, which are integrated by neurons. And that is the end of my today's session. My references for this entire series, especially for this first session, are primarily two books, Principles of Neuroscience by Eric Kandel and co-authors, and a very accessible and interesting book, Exploring the Brain, by Mark Baer and co-authors. So I'm going to stop here. Thank you for your patient listening. A part of this will be dealt with in the third session of this series. Thank you.
Harsha sir, first of all, I would like to ask, I'm extremely sorry that because of I was driving, in between there used to be, I was never getting the signals. That in between I used to leave the webinar and again rejoin and all those things. Sorry for that. And
no problem at all, sir. No problem at all.
And in the meantime, I had almost at least 70% lecture I learned almost. Thank you. May I ask participants to, if any questions are there, you can put it in the chat box or you can ask it directly, Dr. Harsha. And actually, with respect to the this first topic, neuronal signaling, actually, even during my first MBBS physiology classes or during my DPM time or my MD time, I had a very tough time. It is very difficult to understand. Appears very easy, in fact, but it needs frequent rehearsals and all those things. Actually, because of COVID, in the last 1 week, I am working with this oxygen consumption, refilling of the liquid medical oxygen, and now I am working more with this anesthetist and biomedical engineer. Now I am learning how it consumes and all. And looking at this COVID scenario and all those things, so neurophysiology is one of the very, very important topic for any disorders to understand better and to further to plan it out. Especially right now in the COVID moment, how oxygen requirement is needed and all. For that, both neurophysiological and neurochemical changes we need to understand a lot. So in this aspect, I think this is a very, very important topic to understand neurophysiology related issues with the special emphasis on psychiatric topics. So may I now request postgraduates to ask questions or put the questions in the chat box.
Yes, questions will always be welcome in the future sessions too. I understand that this might have been a bit of a a fly-through session,
Yes,
but given the the time constraints, one has to plan for because we want to spend more time on the higher levels of understanding, in the sense that network and systems levels of understanding. We thought we'll keep only one session for this, and therefore we had to kind of keep it quite rapid. It was a kind of a rapid fire session. But if time is available, this would be the right time for students to quickly review some of the basic concepts. Even most psychiatry textbooks do provide these fundamentals in a very nice manner. I've seen that. So a quick refresher of this would really help in moving on with the series.
Yes, actually, Riddhi has commented saying that you made our concepts very clear and it was a refreshing course. Definitely, thank you for that. And also, Bhushan has commented like it's a very crisp and good revision of basic concepts. But actually, that it appears very easy when you listen to somebody talking on neurophysiology or neurochemistry, but which needs repeated rehearsals, because many times even I felt it's just like another this research methodology also, same thing. So everything goes very easy when you listen once or when you read, but if it doesn't rehearsal, very difficult to understand the concepts. And based on the concepts, neurophysiology and neurochemical concepts, we can better understand the disease prognosis as well as the management of the thing, especially with respect to rational psychopharmacology, etc. So in this aspect, I would like to request the postgraduates to have at least more rehearsal after going from this topic. So kind of go repeat, do the rehearsal so that you can come up with the questions whichever you have in the next session also. It's not an issue that you have to ask now only. And somebody has commented that with respect to the neurotransmitters, they need more information, sir. So hope Dr. Harsha will definitely give a good feast about on neurotransmitters also. So nothing to worry. So if there are no questions, may I ask Dr. Mahesh Gowda sir to comment on this?
Dr. Harsha, it's been a long time since we heard you, but each time we hear you, each class looks innovative. Your slides, animations are the best. I think you've been like Stephen Stahl trying to teach us neurophysiology here. And your animation are so self-explanatory, and there are no actual bullets or pointers for you to write down any points, but it's a lot of listening. Unless you really put in 100%, you tend to miss out on so many of your discussion that you made. Being clear on this sort of concepts and being so passionate, I think, is what Dr. Harsha is. And I think you've done justice to today's program. It was wonderful having you, Dr. Harsha, here. And I'm sure students and all other people will be willing to wait for the next class and want to learn more. This class is also recorded and it's going to be on YouTube, and it'll be there for the students to access and learn from it a little later also, for the benefit of all the people who could not make it. Thank you, Harsha, for agreeing to be a resource person, and IPSKC is definitely going to be thankful and will remember your contribution towards educating the postgraduate students of Karnataka and elsewhere. Thank you so much, Harsha.
Thank you, Mahesh, and thank you, Dr. Somshekar. Of course, the neurotransmitters, I think one of the students, Vani, has asked, can we cover more about neurotransmitters? What we'll do is we will discuss specific neurotransmitters in the context of what we're going to discuss in the coming sessions. We probably will not have time for a separate session on that, but we will try and in the context of various things we'll touch upon that. Thank you so much. I think what Dr. Somshekar was saying is very, very true. Revision and rehearsal is the key. The only reason that I can talk with some amount of confidence about these things is because over probably the last 20 years, I've been talking about this repeatedly. So, teaching this to students repeatedly is the only reason why I can do this with some amount of confidence. So, Dr. Somshekhar is bang on there. The only way to understand this is self-study and revision. There's only a little bit, all that I aim to do in my classes, I keep repeating this, all that I aim to do in my classes is basically to, create interest in this. One cannot teach this in 1 hour or even in 20 hours. One can only create interest and kind of point out the broad principles. But the only way to learn this is through self-study. So I'm not going to shy away from saying that you will not be learning. You'll be not be really be learning much from my sessions. My only goal and hope is to create some interest in these topics. Thank you.
Okay. On behalf of Indian Psychiatric Society, I would like to thank today's speaker, Dr. Harsha Halahalli. Thank you, sir. And also, I thank Dr. Mahesh Gowda, the Telearaga team coordinator, as well as Icon Laboratory, Mr. Sriram, for giving us IT platform. And I would like to thank all the participants. And I would like to inform you that there are 2-3 physiology professors also joined the session today, sir. Especially from my college, Dr. Samta has joined. Thank you, madam. Thank you very much. So thank you, one and all. So we'd like to end the session, today's session, and we'll again see you all on next week at 7:30. Thank you very much. Good night, one and.
Thank you, everybody. Good night.
Thank you. Thank you. Good night. Stay safe.
Thank you. Thank you, Sriram.
Thank you, sir.
Sriram, thank you.
Very much, sir.
Sriram?
Yes, sir. Thank you, sir.
Yeah, thank you. You can end the session.
Okay, sir.
Hello. Yes, sir. One patient we have admitted, sir, now. Sir, 66-year-old male.